Hey guys, so today we're going to be talking about photosynthesis by that high school tutor. I have a little stuffy nose today, so bear with me. So, photosynthesis has three stages or three parts. It starts off with capturing light, then goes into the light reactions, and then finally ends off with the Calvin cycle. Now I have the photosynthesis equation here for you guys. We start off with our reactants. Our 6 carbon dioxides, our 6 water molecules, and sunlight. Sunlight is not usually written into a chemical equation, but it is there and it is a vital part in starting in photosynthesis. These reactants come together in different stages of the cycle to create 1 glucose molecule and 6 oxygen gas molecules. So let's start off with capturing light. It begins with the pigment in chloroplasts, which are a type of plastid. Plastids are an organelle in a cell that does photosynthesis. The pigment in most plant cells and most cells is chlorophyll. And these pigments / chlorophyll absorb lights from the spectrum and reflect certain colors. And these pigments are located in the thylakoid membrane. This right here, this disc right here, that's what the thylakoid is. As you can see here, the pigment is absorbing all these colors except the green. That's why whenever you look outside and it's a nice sunny day, you see green trees and green plants, mostly because of the pigment chlorophyll in their leaves. So, before we move on and before I explain even more, I have this diagram here for you to show you. If you need to reference this anytime while I'm talking, just go back and take a look. But I will be explaining it in process as well, just for purpose of the next slide. So the light reactions. I've brought back our photosynthesis equation, and we are going to be using in the light reactions the 6 water molecules to make 6 oxygen gas molecules. Inside of the pigment, these electrons that you saw get energized, and the electrons come from water being split by an enzyme. Then these energized electrons move through the electron transport chain, which is just what you saw before, the thylakoid membrane. While the electrons move through, some of the energy is lost to transport hydrogen ions from the thylakoid membrane to the lumen, or you can say the thylakoid space, whatever floats your boat. This uses active transport. The hydrogen ion gradient is used to make ATP using the ATP synthase. Electrons get re-excited and provide energy to make NADPH, which is another important molecule. Okay, so that was a lot, wasn't it? So I will be explaining it to you in diagram form. So we have our water molecules. We need 2 of them to make 4 hydrogen ions and 1 oxygen gas molecule and 4 electrons. Now, we know that electrons have a negative charge, and this ion has a positive charge. We have our water splitting enzyme. Oh, just to mention, this is our thylakoid membrane, because we're in a thylakoid. We have our water splitting enzyme right here next to the pigment. It takes the water in here and then splits it apart to make these 4 products. The 4 electrons, the oxygen gas, and the 4 hydrogen ions. Now, how did we get our product? Well, this oxygen gas is already made. So, this oxygen gas just leaves the cell and leaves the plant back into the atmosphere. And since it's a gas, it can pass through the membrane easily. So, we will talk about the hydrogen ions later, but let's first go here. Now, you're thinking, well, I already made the product, so why do we have to go through this whole step if we already made oxygen gas? Why can't we just have a water splitting enzyme that does that? Well, we need these electrons from the water to power the next stage in photosynthesis, the Calvin cycle. So, as I said before, the water splits into these 3 parts. The electrons, they move through the pigment. chlorophyll in this case, that absorbs the light, remember? So, because of this, these electrons get energized inside of this pigment. That's what the red line is representing, the energy levels of the electron. So, let's imagine this little dot is an electron, okay? Little red dot. It moves up and it's really, really, really excited in the pigment. And it keeps being excited as it moves through, carrier proteins. Then you hit a block. Okay, so now we need to use the electron's energy to transport these hydrogen ions into the thylakoid space / lumen. Now, it requires energy to do that because this is a form of active transport. You're moving things from a low concentration to a high concentration, or in this case, against the concentration gradient. To do that, you require energy. Instead of using energy from usually ATP, we are using the energy from this electron to transport these molecules. As you can tell and see, the energy levels of the electron has gone lower into another carrier transfer protein, up into another pigment, another chlorophyll, maybe another one that also absorbs light again. But you see the red line going up again. That's because the electrons are energized again because of the light. And then we reach an enzyme that puts NAD plus hydrogen ions to make NADPH. It uses the energy from these electrons to combine these two to make NADPH, which is going to be really important in the next stage of photosynthesis. Now, we didn't talk about the hydrogen ions. Why do they need to come here? So, we use this hydrogen ion gradient to make ATP. So how does that happen? Like we said before, the hydrogen ions in here use this hydrogen ion pump protein to transport these hydrogen ions from a low concentration to a high concentration. Now, we need to bring these hydrogen ions down into this ATP synthase. It says ATP producing carrier protein, but it's most commonly known as ATP synthase. So, this is a type of passive transport. This is active, this is passive. Because we're going down the concentration gradient. So as it's moved through, it produces ATP by using this gradient. That's most of it. So let's do an overview. Water is split to make 4 hydrogen ions, 1 oxygen gas molecule, and 4 electrons. So this leaves the cell, and we use this hydrogen ions back into this area. The electrons get energized by the pigments from the light, but then they lose energy whenever a hydrogen ion pump is there to transport these hydrogen ions down. And then the electrons get re-energized by the light and then sent through an enzyme to make NADPH. You need 2 electrons to make NADPH to form that covalent bond. Okay, so that's the light reactions, the second stage of photosynthesis. Now we talk about the Calvin cycle. I brought back our photosynthesis equation to show that we will not be using water and oxygen because we use that in the light reactions. Now we are going to use carbon dioxide and glucose. Here's a general picture for you. You see the ATP and the NADPH that we made from the previous cycle, the light reactions. And our final output is glucose. Like I said before, these molecules, these energy-carrying molecules, are used in this Calvin cycle. And it occurs in the stroma of the chloroplast. You might have saw in the last slide that there was something in the left-hand bottom corner that said stroma. That's basically the liquid inside of a chloroplast. Now, this is a series of enzyme-assisted chemical reactions. CO2, or carbon dioxide, is incorporated into organic compounds like RUBP, which is something that you'll see later. Again, organic compounds is things that have carbon in it. Rubisco is an enzyme that catalyzes or starts the Calvin cycle. So here's a picture for you. I really like this picture because it really shows you. So let's go. So we have, according to the picture, this cycle has to be done twice to produce one glucose molecule, according to the picture. But in reality, you usually only use one carbon dioxide per cycle. So you would actually have to do the cycle 6 times in order to get one glucose molecule. But let's not worry about that for now. Let's do what we see. So RUBP is a 5 organic compound, 5 carbon organic compound that starts off this reaction. This is our baseline for the reaction. As you can see here, there are 2 phosphates at the end, and we have 3 of these. We also have 3 carbon dioxides. So remember the Rubisco enzyme we were talking about in the last phase? So this is coming back again. We use Rubisco to combine a carbon dioxide with the RUBP. As you can see here, we have the carbon attached here with maybe oxygen molecules hanging out from here. Yes. So the carbon from the carbon dioxide is attached to the RUBP. Now, another enzyme at the arrows splits it up, making it phosphoglycerate. You don't need to know these names, it's just very confusing. But you see that one phosphate is gone because we split it, that's why. Now, we have to use the ATP from the last stage. We need to use 6 ATP to energize, to add a phosphate to the phosphoglycerate. And in that process, we get ADP, which can be recycled again to make ATP. Now, there's another enzyme here that turns the biphosphoglycerate into G3P. But instead, we need to add more energy to make sure that this can be converted into G3P. So instead of using ATP, we use NADPH. Now the NADPH brings energy into this molecule, and since it already has energy from the NADPH, it doesn't need the phosphate anymore. So it gets rid of the phosphate, and it can be reused later in other processes. Now, we have our G3P here. One of these 6 G3P molecules will go out of the Calvin cycle to become glucose. But the other 5 G3P are reused to make RUBP, to restart the cycle all over again. As you can see here, the G3P uses ATP to combine it back into RUBP. Like I said before, this has to be done twice to make sure that we get one glucose molecule, according to the picture. Okay, here's another picture for you if that one was a little too confusing. It explains it pretty well. Like I said before, the carbon dioxide molecule goes here, makes this 3 carbon compound with ATP and NADPH, and makes G3P. One of these G3Ps becomes a glucose, and then the rest are recycled. So, fill in the blank. Pause this video and tell me if you can fill in the blank. Okay, so the first stage of photosynthesis is capturing light, as you can see here. The thylakoid is capturing this light. Then we have our light-dependent reactions in the thylakoid. And then we have our Calvin cycle. I think this is a really good drawing because it really shows what the products from each cycle are and how they're used in correlation with each other. So, that is the end of our video on photosynthesis. If you have any questions, please put them in the comments and I'll get back to you. Bye!